Right now, in those tiny, microscopic gaps between your brain cells, there's a constant, super high-speed conversation happening. And today, we're going to decode the chemical language of that conversation and meet the very first messenger ever discovered, acetylcholine. Now, this might look pretty chaotic, right? But what you're seeing is actually one of the most organized conversations in the entire universe. This is a synapse, that tiny gap where one neuron talks to another. Seriously, every thought, every memory, every single movement you make, it all begins with a message passed across this tiny space, a space that's less than a millionth of an inch wide. So, if this is a conversation, what language are these neurons speaking? Well, it's not just electrical sparks flying around. The real language of the synapse, it's all chemical. And that language is spoken by these incredible molecules called neurotransmitters. You can think of them as the brain's own couriers, carrying signals across that synaptic gap. And it's a really rich language with over 100 different types of these messengers, and each one has its own unique job to do. We can basically group these messengers by their size. You've got the large ones called neuropeptides, and then there are the smaller, faster-acting ones. And this small molecule group, well, it includes some heavy hitters you've probably heard of, like dopamine and serotonin. But today, we're focusing on a real pioneer in the field, acetylcholine, or ACH for short. So let's meet our star for today, acetylcholine. What makes this one so special? Well, it actually holds the title of being the very first neurotransmitter ever identified by scientists. It's the OG, the original chemical messenger that gave us our first peek into how our nervous system really works. And believe me, ACH is a serious multitasker. It's the signal that tells your muscles to contract, it's the brake your nervous system uses to slow down your heart rate, and deep inside your brain, it's a key player in learning and in memory. So from doing a bicep curl to just recalling someone's name, yep, ACH is the molecule that's making it happen. To really, truly understand acetylcholine, we've got to see it in action. So we're going to take a little guided tour of its entire life cycle right there at the synapse. We'll see how it's born, how it delivers its message, and how it gets cleaned up. And this all happens in just a fraction of a millisecond. It's wild. All right, so this right here, this is the whole arena for our story. Up top, you've got the presynaptic terminal, that's the sending neuron. Down below is the postsynaptic cell, that's the receiver. And that tiny little gap in between, that's the synaptic cleft. That is where all the magic happens. The whole process happens in 5 lightning-fast stages. First, the neuron builds the ACH from raw materials. Simple enough. Then it packages it all up into these tiny bubbles called vesicles. When the signal comes in, it releases the ACH into the gap. The ACH then zips across and binds to receptors on the other side, delivering the message. And finally, and this is crucial, the system immediately starts the breakdown and recycling process to get ready for the very next signal. And this cleanup part is just astonishingly fast. I mean, the signal has to be turned off just as quickly as it's turned on, right? That's the job of an enzyme called acetylcholinesterase, or ACHE. Think of it as a molecular cleanup crew that's on total overdrive. It breaks down something like 5,000 molecules of ACH every single second. It's that incredible speed that allows us to have such precise, rapid control over things like our muscle movements. So what happens if you jam up that recycling machine? Well, the consequences are absolutely devastating. Things like nerve agents, sarin gas for example, they work by blocking ACHE. Without that cleanup crew, ACH just floods the synapse, constantly hammering the receptors over and over. The receiving cells get completely overwhelmed and just shut down. The result is total paralysis. It's a terrifying look at just how vital this one cleanup step really is. Okay, so we've met the messenger, our key, ACH. Now let's take a look at the lock it's designed to open. Meet its most famous target, the nicotinic receptor. And yep, you guessed it. It gets its name because it's also activated by nicotine from tobacco. Just look at this thing's complex structure. It's not one solid piece of protein. It's more like a finely tuned machine built from 5 separate protein subunits. You can see them here, all arranged in a circle, and they create a channel or pore that runs right down the middle. Now, this kind of structure has a special name, a ligand-gated ion channel. Let's break that down because it sounds more complicated than it is. Ligand is just a fancy word for any molecule that binds to a receptor. In our case, that's our messenger, ACH. So this is basically a gate that only opens for ions when the right chemical key, ACH, fits perfectly into the lock. And this lock is really specific. It needs not 1, but 2 ACH molecules to turn the key.
When both of them bind, the entire structure literally twists open, unblocking that central pore. This allows a flood of positive ions to rush into the cell, which then excites it and carries that signal forward. It's so precise.
This whole system is so precise and so essential to life that, of course, it's become a target. Over millions of years of evolution, predators and prey have developed these potent chemical weapons that attack this exact process. And we're also going to see what happens when the body's own immune system gets confused and decides to attack itself. Meet the banded crate. Its venom contains a molecule called alpha-bungarotoxin. Now, this molecule is shaped almost perfectly to fit into that ACH receptor. But here's the twist. Instead of opening the gate, it just jams the lock. It binds so tightly that ACH can't get in at all, completely shutting the signal down and causing paralysis. And nature is just full of these examples. I mean, look at this cone snail. It actually hunts fish by firing a venomous harpoon-like tooth. Its venom is a cocktail of hundreds of different toxins, and many of them, called conotoxins, also work by blocking these same exact ACH receptors, instantly immobilizing its prey. It's a literal biological arms race, all centered on this one vital synapse. But the threat doesn't always come from the outside. Sometimes the most devastating attacks are a case of friendly fire, when the body's own immune system makes a terrible, terrible mistake. This data right here, it tells the whole story. What you're looking at is an EMG, which is a recording of muscle activity. That top graph shows a healthy response. See? A strong, steady signal. But the bottom one, that's from a patient with a disease called Myasthnia gravis. It's totally different. The signal starts, but then it just immediately fades away. The muscle gets tired almost instantly. So what's going on here? Well, the cause is an autoimmune disease called Myasthenia gravis. In this case, the body's own immune system mistakenly attacks and destroys its nicotinic ACH receptors. With fewer and fewer mailboxes available to deliver the message to, the signal gets weaker and weaker, kind of like a radio station just fading out. And that is what causes the profound muscle weakness that we saw in that EMG. So we're left with this incredible paradox, aren't we? The very same molecular machinery that gives us the power to think, to remember, to move, it's so essential that it's also a primary target for nature's deadliest poisons and a tragic victim of the body's own mistakes. I guess it just reveals the amazing power and also the profound vulnerability of that delicate chemical balance that makes us who we are.